Some stories leave behind questions that echo for decades, and the Darrow case is one of them. A single family, a single mark repeated across every child, and a medical mystery that challenged everything we thought we knew about chance and inheritance. Before we begin, where are you watching from? Drop it in the comments. I'd love to see how far this story is reaching. The Darrow children entered the world one after another, each carrying a visible mark that could not be overlooked. On the day of each birth, midwives and physicians noted a similar pattern on the skin, a patch of color that was neither fleeting nor accidental. Birthmarks were not uncommon in newborns, but what set the Darrow family apart was the repetition. Every child bore a mark of the same type, in the same general place. The observation was unusual enough to attract the interest of clinicians who understood how rare it was for multiple siblings to share an identical cutaneous feature. The first task of those who examined the children was classification. All marks of this kind fall into two established categories: vascular, caused by blood vessel anomalies close to the skin, or pigmented, caused by concentrations of melanocytes. This distinction matters because the natural history of the mark, whether it fades, persists, darkens, or spreads, depends on which group it belongs to. Flat pink patches, proliferating red nodules, or brown spots each suggest different mechanisms and carry different implications for health. What made the Darrow case more than a curiosity was the sheer improbability. Many birthmarks are fairly common. Salmon patches, often called stork bites or angel kisses, occur in a significant portion of infants and tend to disappear with time. Café au lait spots also appear regularly in the general population, varying by skin type and ethnicity. Hemangiomas, vascular growths that emerge in infancy, affect roughly 1 in 20 children. With marks as common as these, coincidence is possible, but coincidence becomes statistically less likely when several siblings exhibit marks not just similar in nature but strikingly alike in appearance and placement. The Darrow children's marks seem to exceed the boundaries of chance. Medical investigators, when faced with such a pattern, turn to the frameworks that decades of clinical research provide. Some explanations lie in genetics. Certain studies have described families in which vascular birthmarks, particularly infantile hemangiomas, appear in several members, suggesting a heritable predisposition. These cases rarely follow the clean lines of simple Mendelian inheritance, but instead show complex or multifactorial transmission, with contributions from multiple genes and perhaps from maternal influences during pregnancy. In other families, researchers have found no germline mutation at all, pointing instead to mosaic events, mutations that arise after conception in a subset of cells. These somatic mutations, such as those identified in port wine stains linked to the GNAQ gene, explain why most cases occur sporadically and not in repeated family patterns. The Darrow children's situation prompted investigators to weigh these possibilities carefully. If the marks were due to common types like salmon patches, coincidence could not be ruled out. If they resembled hemangiomas, then familial aggregation became more plausible, especially if risk factors such as low birth weight or prematurity were present in the siblings' histories. But if the marks were port wine stains or other vascular malformations usually linked to somatic mosaicism, then a different mechanism had to be considered, one that might involve gonadal mosaicism in a parent or an unlikely series of independent mutations in each child. To untangle the truth, the clinical pathway followed established steps. Careful documentation was the first, with physicians photographing and describing the marks in detail, their shape, color, and borders, as well as how they changed over time. Family histories were collected, not only from the immediate household but from extended relatives, to see whether similar marks had appeared in earlier generations. When indicated, systemic evaluations were pursued, since certain vascular lesions can signal underlying syndromes involving the nervous system or eyes. Genetic investigations followed, comparing DNA from blood samples to DNA drawn from affected skin, a method that could distinguish between germline inheritance and post-zygotic mutation. As these steps unfolded, the weight of evidence tilted toward explanation rather than mystery. The recurrence of identical marks across siblings was highly unusual, but not beyond the boundaries of what medicine could account for. Whether the explanation would be coincidence, shared prenatal factors, a heritable predisposition, or the subtler mechanism of parental mosaicism remained to be seen. For now, The Darrow children's case stood at the intersection of curiosity and inquiry, a reminder of how visible skin findings often lead to deeper questions about inheritance, development, and biology itself. As the Darrow case drew more attention, the emphasis shifted from first impressions to the disciplined accumulation of data. Clinicians and researchers began to place the family's story against the backdrop of known birthmark epidemiology. Patterns across populations provided a way to determine whether what had happened to the Darrow siblings was an unlikely but random clustering or something that pointed to a deeper biological cause. In large cohorts of newborns, vascular marks are reported with striking regularity. Small salmon patches, for instance, occur in a significant percentage of infants, and many fade before school age. Hemangiomas, though less frequent, are still seen in several percent of births, with risk factors such as female sex, low birth weight, and prematurity consistently documented. Café au lait spots vary in prevalence but can be found in healthy children as well as in those with certain genetic syndromes. These background numbers matter because they establish the probability of recurrence by chance. For 3 or 4 siblings to each present with the same distinctive mark, the odds shrink rapidly, raising the likelihood of a unifying explanation. Attention therefore turned to genetics. Researchers examining families with recurrent hemangiomas have observed patterns suggestive of heritable predisposition. Some pedigrees indicate an autosomal dominant mode, though penetrance is often incomplete, while others suggest multifactorial inheritance. Candidate gene studies and more recent sequencing approaches have revealed possible associations, but no single gene has yet been established as universally causative. The Darrow family seemed to echo these ambiguous findings, an inheritance pattern that hinted at genetics but resisted simple categorization. Other pathways could not be ignored. Somatic mosaicism, the mechanism by which a mutation arises after fertilization in a subset of cells, is well established in certain vascular birthmarks. Portwine stains, for example, are strongly linked to activating mutations in the GNAQ gene, detectable in the affected skin but absent from blood or other tissues. Because these mutations are not in the germline, they are not usually passed on. For multiple siblings to share such lesions would require either a parent with gonadal mosaicism or several independent events. While unlikely, such scenarios are not impossible and have been documented in other conditions. The Darrow children's birth records were examined closely. Their gestational ages, birth weights, and maternal histories were compared for clues. Epidemiological studies show that prematurity and low birth weight increase the risk of infantile hemangiomas, while certain maternal factors, such as multiple pregnancies or advanced maternal age, may also contribute. If these risk factors were shared across the Darrow siblings, then environment and maternal conditions could be at least part of the explanation. Investigators also considered syndromic associations. Café au lait spots, when present in numbers, may suggest neurofibromatosis type 1, a genetic condition that carries a range of other clinical features. Extensive vascular lesions can be linked to neurocutaneous syndromes such as Sturge-Weber. The Darrow children, however, did not show signs of systemic illness at first evaluation, and so their case seemed more likely to represent an isolated dermatologic phenomenon. Even so, vigilance remained necessary, for some syndromic features emerge only later in childhood. To move from conjecture to evidence, genetic testing was planned. Blood samples from the siblings and their parents could reveal whether a germline variant was present. If nothing appeared in the blood, then tissue samples from the birthmarks themselves could be analyzed for mosaic mutations, comparing affected to unaffected skin. This dual approach, germline testing and lesion testing, represented the most rigorous way to distinguish inherited predisposition from somatic change. The investigators knew that each potential explanation carried different implications. If the cause proved to be a heritable variant, then future children in the family might carry the same risk, and genetic counseling would be essential. If the explanation was mosaicism, the recurrence risk would likely be low, though not zero if gonadal mosaicism were involved. If perinatal factors were responsible, then obstetric management could potentially mitigate risks in subsequent pregnancies. The Darrow case, therefore, was not only about solving a scientific puzzle, but also about guiding medical advice for the family's future. In this second stage, the inquiry deepened into the mechanics of probability, genetics, and developmental biology. The children's marks were no longer viewed simply as curiosities, but as windows into processes that determine how blood vessels form, how pigment cells distribute, and how mutations shape visible traits. The family became part of a broader effort to understand why some patterns repeat in biology and others occur only once, and whether visible skin findings can serve as signals of hidden molecular truths. The third stage of inquiry into the Darrow children's condition involved the careful accumulation of molecular evidence. Visual examination and family history had raised critical questions, but modern medicine demands proof at the genetic level whenever possible. Samples were prepared, small biopsies from the affected patches on each child's skin, alongside blood samples from both parents and children. These would allow investigators to determine whether the birthmarks were linked to germline inheritance or to mutations confined to the lesions themselves. The methodology was grounded in established research practices. In recent years, sequencing of lesion tissue has revealed that many vascular birthmarks are the result of somatic mutations. Port wine stains, for example, are frequently associated with mosaic variants in the GNAQ gene, mutations that alter signaling in vascular endothelial cells. Because such mutations occur after fertilization, they affect only a subset of cells and do not appear in blood DNA. This discovery fundamentally changed how clinicians think about recurrence. Port wine stains are typically sporadic and familial patterns are rare. If the Darrow children's marks were of this type, the results from lesion sequencing would be decisive. At the same time, investigators remained aware of studies documenting familial clustering of infantile hemangiomas. In those families, the inheritance appears complex, with some evidence for dominant transmission and other cases suggesting multifactorial risk. No single gene has yet explained all such patterns, but variants in genes involved in angiogenesis and vascular development remain under study. If the Darrow samples revealed a germline variant present in all affected siblings and in one parent, the case would align with this line of research. While laboratory testing proceeded, the clinicians also reviewed the broader literature on recurrence. They noted that gonadal mosaicism in a parent could explain multiple affected offspring, even if blood testing of the parent appeared normal. This mechanism has been confirmed in other genetic conditions and remains a crucial consideration whenever several siblings share a trait unexplained by typical inheritance. For the Darrow family, this meant that even if no mutation was detected in parental blood, the possibility of hidden germline mosaicism remained. The role of perinatal environment was also revisited. Population-level studies have consistently linked hemangioma risk to low birth weight, prematurity, and multiple gestations. The Darrow birth records showed patterns worth considering. Shorter gestational ages and below-average weights were noted in more than one child. Although these factors could not account for identical placement of lesions, they strengthened the hypothesis that maternal and perinatal conditions acted alongside genetic predisposition. As testing advanced, the significance of the findings extended beyond the Darrow household. Researchers saw in this family a rare chance to clarify how visible skin marks might point toward underlying molecular mechanisms. If a heritable variant were confirmed, it would join the growing body of evidence that genetics plays a stronger role in vascular anomalies than once assumed. If instead somatic mosaicism were identified, the case would underscore how postzygotic mutations can create striking similarities without true inheritance. Either way, the results would refine counseling for families who face the anxiety of unexplained recurrence. By the time the Darrow case entered its fourth stage of investigation, the family's experience had become the subject of conferences and detailed reports. Dermatologists, geneticists, and pediatricians each contributed their expertise, seeing in the children's marks not only a clinical puzzle but an opportunity to clarify how visible skin features arise from hidden biological events. The marks themselves remained stable, neither fading nor enlarging, a fact that further guided the line of inquiry. Clinicians recognized that the persistence of these lesions distinguished them from the transient vascular proliferations often seen in infancy. Infantile hemangiomas, for instance, tend to appear within weeks of birth, grow rapidly, and then regress over time, often leaving behind minimal traces. In contrast, congenital vascular malformations, including port wine stains, typically remain unchanged in distribution and may even darken or thicken with age. The Darrow children's lesions, unchanged over several years, were more consistent with this second category. This narrowed the likely mechanisms involved and focused attention on somatic mosaicism as a leading possibility. Yet, the identical appearance across siblings resisted easy categorization. If each mark was due to a random postzygotic mutation, then their recurrence in multiple children seemed improbable. Investigators returned to the concept of gonadal mosaicism in a parent. If a mutation arose early in one parent's germ cells, it could be transmitted to multiple offspring without being detectable in blood or somatic tissues. While rarely proven in practice, this mechanism had been documented in other conditions and offered a coherent explanation for the Darrow family's pattern. The question then shifted from whether the marks were inherited to how the risk could be measured. Families in similar situations often sought clarity. What were the chances that future children would carry the same mark? If the cause was a germline mutation with dominant inheritance, recurrence risk could be as high as 50%. If gonadal mosaicism was at work, the risk was lower but still real, though difficult to quantify without direct testing of germ cells. If instead the marks were coincidental or influenced by shared prenatal conditions, then the recurrence risk might be minimal. Establishing which of these applied required careful interpretation of genetic results and transparent communication with the family. Parallel to the genetic inquiries, epidemiologists placed the Darrow family in the context of population data. They examined registries, cohort studies, and case series that documented familial recurrence of vascular lesions. Such studies confirmed that while most cases were sporadic, clusters within families did occur and were more common than once assumed. This contextual evidence supported the idea that what happened in the Darrow household was not beyond the boundaries of medical expectation, but rather a reflection of the biological diversity that underlies human development. The narrative of the case thus expanded beyond the immediate question of why these siblings shared the same mark. It became a demonstration of how medicine balances probability and mechanism, using every tool available to distinguish coincidence from causation. The Darrow children's marks, once seen as curiosities, had now been transformed into a model case through which clinicians could explore the interplay of genetics, mosaicism, and environment. The marks on their skin were constant, but the meaning attached to them evolved with each layer of investigation. What began as a visible similarity among siblings was now situated within the broader scientific effort to understand how genes, cells, and developmental events leave traces on the body's surface. The Darrow family's story reminded observers that the skin, so often viewed as a barrier, can also serve as a map of hidden biological processes. The attending dermatologists created a schedule of yearly examinations, with standardized photographs taken under controlled lighting. This approach allowed precise comparison of color intensity, border definition, and lesion size over time. By the third year of observation, the Darrow marks showed a striking consistency. Their borders remained sharply defined, their color stable, and their placement unchanged. Unlike hemangiomas, which often enlarge and then regress, these lesions had an unchanging persistence that pointed toward structural vascular malformations rather than proliferative tumors of infancy. At the same time, specialists remained alert to systemic associations. Certain vascular lesions, when extensive or located on specific parts of the face, may signal involvement of deeper tissues such as the brain or eyes. The Darrow children, however, showed no neurological symptoms, no ocular complications, and no developmental delays. This absence of systemic involvement provided reassurance that their condition was confined to the skin. Still, vigilance continued, since syndromic associations can occasionally emerge later. Parallel to clinical follow-up, laboratory teams pursued molecular testing of the skin samples. The sequencing of affected tissue became the central task, seeking mosaic mutations known to drive vascular malformations. Genes such as GNAQ, GNA11, and others involved in signaling pathways were the primary targets. Blood samples from the children and their parents served as controls, allowing direct comparison of germline and lesion-specific DNA. The aim was to detect whether a somatic mutation explained the marks, or whether a heritable variant underpinned the pattern. The preliminary findings were inconclusive. No mutations appeared in the blood DNA, which suggested that the marks were not caused by a readily identifiable germline variant. However, low-level variants detected in the lesion tissue raised the possibility of somatic mosaicism. Because such variants may be present in only a fraction of cells, their detection requires sensitive methods, and sometimes results remain ambiguous. For the Darrow case, this ambiguity meant that no single explanation could yet be confirmed. Researchers widened the scope of inquiry. If the lesions were indeed mosaic, then their recurrence across siblings could be explained only by gonadal mosaicism in a parent. This hypothesis aligned with the consistent appearance of the marks, but it was nearly impossible to prove without direct access to parental germline tissue, a form of testing not feasible in practice. Thus, the case rested at an intersection of probability and inference, where clinical patterns and genetic evidence pointed toward a mechanism but left definitive proof out of reach. In the meantime, the Darrow children grew. Their marks became a part of their identities, features as familiar as hair color or eye shape. Family photographs documented not only the similarity of the marks but also the normal development of the children in every other respect. This reality reinforced an important point. While birthmarks may carry medical significance, they do not necessarily diminish health or function. For the Darrow family, the marks were both a source of scientific scrutiny and a simple familial trait, visible reminders of the mysteries still held by human development. The long-term monitoring of the Darrow siblings provided a rich dataset. Each year of life added more information to the timeline of their condition. The marks, observed under controlled conditions, showed neither enlargement nor regression. Their pigmentation remained stable, with only minor shifts in shade that could be attributed to natural changes in skin tone as the children aged. Their location was constant, unaffected by growth or development. This stability contrasted sharply with the behavior of hemangiomas, which typically show a dynamic course, and strengthened the conclusion that the Darrow lesions were structural vascular malformations rather than proliferative vascular tumors. In conferences where the Darrow case was discussed, the persistence of these lesions became a key point of emphasis. Port wine stains caused by somatic mutations in genes like GNAQ are the prototypical example of vascular malformations that endure throughout life. Studies had already shown that such mutations occur postzygotically, confined to a subset of cells in the affected region. In most patients, these mutations arise sporadically, explaining why the lesions rarely appear in more than one family member. Yet here, in the Darrow siblings, the pattern repeated across several children, forcing experts to grapple with the low probability but biologically plausible mechanisms that might explain it. One mechanism under close scrutiny was gonadal mosaicism. If a parent carried a mutation only in their germ cells, they could transmit it to multiple offspring without showing any sign themselves. This hypothesis fit the Darrow data. The children had marks that were indistinguishable, yet no parent carried visible lesions. Gonadal mosaicism had been documented in conditions such as Duchenne muscular dystrophy and osteogenesis imperfecta, where unaffected parents produced multiple affected children despite negative blood tests. Applying this framework to vascular malformations was a logical extension of established genetic principles. Another mechanism remained in discussion: the possibility that shared perinatal conditions had acted as a risk amplifier. Research had consistently linked low birth weight and prematurity to a higher likelihood of vascular anomalies. The Darrow birth records showed that several of the siblings were born at earlier gestational ages than average, with lower than expected weights. While such factors could not directly produce identical marks, they could increase the baseline risk for vascular lesions, making recurrence more likely in a family where maternal factors remained consistent across pregnancies. This line of reasoning emphasized the interplay of genetic predisposition and environmental triggers, a theme increasingly recognized in many areas of medicine. The laboratory findings continued to fuel debate. Sequencing of lesion tissue revealed low-level variants consistent with somatic mosaicism, but the sensitivity of detection left room for uncertainty. The absence of any clear germline variant in blood testing suggested that the mutations were not inherited in a straightforward manner. This left the research community in a state of cautious interpretation, confident that mosaicism played a role, yet aware that mosaicism alone could not explain recurrence in multiple siblings without invoking gonadal involvement in a parent. Beyond the immediate family, the Darrow case began to shape broader research agendas. Teams studying vascular anomalies took it as evidence that familial recurrence, while rare, could offer unique insights into the timing and mechanisms of mutation. If gonadal mosaicism was confirmed, it would demonstrate that somatic mutations traditionally thought to be isolated events could, under certain conditions, cross into the realm of heritability. This possibility had profound implications for genetic counseling, as families with apparently sporadic lesions might in fact face recurrence risks not previously acknowledged. The Darrow children themselves continued to develop normally. Their marks, once the focal point of clinical concern, had become familiar features of their appearance. They did not hinder growth, cause functional problems, or signal deeper systemic disease. For the family, this provided reassurance, even as the scientific community continued to parse the meaning of the marks at a molecular level. The children's everyday lives unfolded like those of their peers, while their skin quietly carried the answers to questions about inheritance and development that remained just beyond full resolution. At this stage, the Darrow case illustrated both the power and the limits of modern genetics. The power lay in the ability to extract molecular data from skin tissue, to detect low-frequency mutations, and to connect visible features with precise biological mechanisms. The limits were equally clear. Certain mutations remained elusive, gonadal mosaicism could not easily be proven, and the boundary between chance and causation remained blurred when recurrence appeared in families. The Darrow children stood at that boundary, their marks both a scientific challenge and a human story. As years passed, the case was increasingly cited in reviews and discussions about birthmarks and inheritance. It was used as a reminder that even common features, when repeated in unusual ways, demand careful investigation. It highlighted the need for long-term follow-up, not only to monitor health but also to gather data that could inform future families and refine clinical guidelines. In this sense, the Darrow case was more than an isolated anomaly. It was a contribution to the collective understanding of how biology writes itself onto the skin. These registries, compiled from hospitals and research centers, cataloged thousands of cases. They showed that most birthmarks occur sporadically, but a small fraction recur within families. Familial recurrence rates of infantile hemangiomas, for instance, had been estimated to occur in about 10% of cases, suggesting a partial heritable component. Port wine stains, by contrast, almost always occurred as isolated events, explained by somatic mosaic mutations. Yet, scattered case reports did describe multiple siblings with port wine-like lesions, raising the possibility that what appeared impossible could, in fact, occur under rare conditions. The Darrow children's data were placed alongside these records. Their marks matched the characteristics of vascular malformations more than those of hemangiomas. They were congenital, persistent, and did not follow the rapid growth and regression cycle of proliferative lesions. This comparison narrowed the explanatory framework further. If they were malformations, then the central hypothesis remained mosaicism, but with recurrence suggesting gonadal involvement in a parent. Genetic counseling sessions with the Darrow parents became increasingly significant. Counselors explained the range of possible mechanisms, the likelihood of recurrence in future children, and the uncertainty that lingered because direct proof of gonadal mosaicism was unattainable. They emphasized that while the marks were unlikely to cause health problems, the scientific uncertainty meant recurrence risk could not be dismissed. The parents were faced with a new kind of knowledge. Their children were healthy, but their family had become part of a story that medicine could not yet fully explain. Meanwhile, advances in sequencing technology offered new opportunities. Deep sequencing techniques allowed detection of mutations at frequencies lower than 1% of cells, a sensitivity not available when the Darrow case first came under review. Reanalysis of the tissue samples using these newer methods identified faint but reproducible signals in affected skin consistent with low-level variants in genes involved in vascular signaling pathways. The evidence was not conclusive, but it reinforced the mosaic hypothesis and pointed toward a biological basis rather than pure coincidence. The broader medical community took notice. In symposiums and published reviews, the Darrow case was cited as a model of how rare family clusters can challenge assumptions about sporadic conditions. It highlighted the importance of collecting longitudinal family data, performing tissue-level genetic analysis, and remaining cautious in genetic counseling when recurrence occurs without obvious germline findings. In effect, the Darrow family became a case study not only in dermatology but also in how medicine handles the tension between probability and mechanism. For the children, daily life continued without disruption. Their marks remained visible, but their health remained otherwise unaffected. In classrooms and playgrounds, the features that had sparked scientific investigation were simply part of their appearance. Yet in clinical circles, the same features were symbols of unresolved questions about how mutations arise and repeat across siblings. This duality, ordinary family life alongside extraordinary scientific scrutiny, was a hallmark of the Darrow narrative. By this stage, the inquiry was no longer about finding a single definitive answer but about charting the edges of current knowledge. The Darrow family illustrated how cases that do not fit neatly into existing categories can stretch the boundaries of science, compelling clinicians to refine diagnostic pathways and genetic frameworks. The visible birthmarks of the children had become markers of something larger. The ongoing effort to connect clinical observation with molecular biology, to bridge the gap between what can be seen and what can be proven. The Darrow case was now frequently discussed in genetic and dermatological conferences as a teaching example. It exemplified the challenges of distinguishing between chance and causation, and it offered a narrative thread for exploring gonadal mosaicism, somatic mutations, and the limitations of current diagnostic methods. Unlike cases where dramatic symptoms push research urgency, the Darrow situation was subtle. The only anomaly was the shared presence of marks. Yet, precisely because of its subtlety, it forced clinicians and scientists to confront the gaps in knowledge about how certain mutations arise and repeat across siblings. By this point, technological advances had changed the landscape of medical research. Ultra-deep sequencing methods and single-cell analysis had become more accessible, allowing investigators to revisit old samples with new tools. Skin biopsies taken from the Darrow children years earlier were reanalyzed. This time, evidence of low-level somatic mutations was clearer. Variants consistent with alterations in vascular signaling genes appeared within the lesions but were absent in unaffected tissue. These findings, while still not definitive, aligned with the theory that each lesion stemmed from a mutation confined to a subset of cells. The persistence of identical marks across siblings continued to raise the same critical question. How could separate mosaic events replicate so precisely? The prevailing interpretation remained gonadal mosaicism in one parent. Although direct testing of germline cells was not possible, the statistical unlikelihood of identical independent mutations, coupled with the consistent phenotype across children, supported this explanation. The Darrow case thus became a touchstone example cited in genetic literature as indirect evidence of gonadal mosaicism manifesting through vascular malformations. The family's contribution extended beyond the research sphere. In genetic counseling, the Darrow example was used to illustrate conversations about uncertainty. Parents in similar situations often asked what the presence of a vascular mark in one child meant for future children. The Darrow data provided a framework. Recurrence was possible, especially when marks appeared in multiple siblings, but predicting risk required humility, acknowledging the limits of current science. For the Darrow siblings themselves, adolescence brought new dimensions. The marks, once accepted as part of childhood identity, became features around which questions of self-image and social perception arose. This highlighted another lesson for clinicians. Medical explanations are only part of the story. Birthmarks, regardless of etiology, intersect with psychosocial development, influencing how individuals see themselves and how they are perceived by others. In this respect, the Darrow case reminded the medical field that clinical follow-up should address not only biological questions but also the human experience of living with visible differences. The first major element was the consolidation of long-term clinical data. The photographs taken annually from infancy through adolescence were collated into a timeline. This collection demonstrated, with remarkable clarity, the stability of the marks. They had grown proportionally with the children's bodies, maintaining consistent size relative to body surface area, and their pigmentation remained essentially unchanged. Dermatologists presented this sequence at conferences as evidence that congenital vascular malformations of this kind do not regress and rarely evolve into more complex lesions, provided no systemic conditions are present. The Darrow dataset thus became a visual and empirical record supporting established theories while also documenting a rare familial recurrence. Parallel to clinical consolidation was the maturation of molecular inquiry. Tissue samples archived during the children's early years had been preserved in biobanks, a foresight that allowed them to be re-examined as sequencing technologies advanced. By this stage, whole genome sequencing at single-cell resolution was possible, a technique unthinkable when the case first entered medical attention. These reanalyses revealed more than earlier testing could. Within the vascular tissue of the lesions, researchers identified somatic mutations in genes involved in endothelial signaling pathways, variants known to drive abnormal blood vessel formation. Importantly, these mutations were absent from the children's blood and unaffected skin, confirming mosaicism as the operative mechanism. Yet even with this discovery, the central mystery remained. Why had all siblings developed nearly identical marks? Mosaic mutations are typically random, confined to one individual, not replicated across multiple siblings. The prevailing explanation continued to be gonadal mosaicism in one parent. The logic was simple but compelling. If a mutation occurred early in the development of one parent's germ cells, then multiple gametes could carry the same variant, leading to recurrence in several offspring. The Darrow family represented a rare, almost textbook example of this process, one that textbooks previously described in theory but rarely illustrated with such clarity in practice. The implications for genetic counseling were significant. Until the Darrow case, gonadal mosaicism had been acknowledged primarily in conditions with overt systemic disease. Here, the family provided evidence that even benign congenital features could follow the same mechanism. Counselors began to use the Darrow example in consultations with other families facing recurrence of unusual skin features. It was a lesson in humility, that even visible marks without health consequences could trace back to complex and poorly understood genetic events. Epidemiologists also used the Darrow data to refine statistical models. They re-examined population-based registries of vascular anomalies, calculating the probability of identical lesions appearing independently in multiple siblings. The estimates confirmed what intuition already suggested. Such an outcome was vanishingly rare without a shared underlying cause. The Darrow family thus became a statistical anchor, a case against which population probabilities could be tested and recalibrated. The Darrow family's cooperation also underscored the importance of trust between researchers and participants. For nearly 2 decades, they had agreed to examinations, biopsies, photographs, and interviews. They understood that while no single answer might emerge, their participation advanced science incrementally. In return, the medical community treated their case with respect, ensuring confidentiality while using anonymized data to inform broader understanding. This reciprocity became part of the story, demonstrating how long-term case studies depend on relationships as much as on technology. The broader research community integrated the Darrow findings into discussions of developmental genetics. The case was cited in reviews that explored how early mutations could lead to visible traits, how somatic mosaicism can blur the line between inherited and sporadic conditions, and how recurrence in families could be explained without invoking coincidence. In this sense, the Darrow children were not just patients. They were contributors to a larger effort to map the hidden architectures of biology. Their story became less about an answer and more about a process. The process of science confronting its own limits and steadily expanding them. The marks on the Darrow children's skin were unchanged, but the meaning attached to them had shifted profoundly. What began as a mystery in a small family had become a cornerstone example in the study of mosaicism, inheritance, and the complex interplay between chance and causation. When the Darrow children reached adulthood, the marks that had first brought them into medical attention remained unchanged. Each lesion had followed the body's natural growth, preserving its shape and proportion with the child's size. What began as faint patches of discoloration visible at birth were now mature vascular stains, clearly defined and stable, as constant in their presence as any inherited physical trait. For the physicians who had followed the family over nearly 2 decades, this constancy provided rare, invaluable documentation of a phenomenon that textbooks described but rarely illustrated with such completeness. The long arc of follow-up was central to the scientific weight of the case. Each year, the family had agreed to detailed clinical evaluations, photographs taken under identical conditions, dermatoscopic examinations, sometimes additional imaging when new techniques became available. The accumulated record stretched from the first days of life to the threshold of adulthood. Taken together, it formed one of the most comprehensive visual archives of congenital vascular lesions in living memory. It demonstrated with precision that these marks did not regress, that they did not alter their position, and that they persisted as stable vascular malformations rather than proliferative tumors. The Darrow siblings' bodies became, in effect, case studies in continuity. The preserved samples from early biopsies gained new relevance as sequencing technology advanced. The first attempts at DNA analysis in the children's infancy had returned inconclusive results, limited by the sensitivity of methods available at the time. But as high-depth sequencing and single-cell analysis became routine, researchers revisited the Darrow material. Skin sections preserved in paraffin were carefully reprocessed, their DNA extracted and amplified. With new methods, faint but reproducible signals emerged. Mutations in genes controlling endothelial signaling, mutations known from other studies to trigger abnormal vascular development. The mutations appeared only in the affected skin, absent from blood and unaffected tissue. This finding confirmed that the Darrow marks were products of somatic mosaicism. The lesions arose from mutations that had occurred after conception, confined to specific populations of cells. Yet the recurrence across siblings still required explanation. The likelihood of identical somatic mutations arising independently in each child was vanishingly small. The investigators turned again to the theory that had hovered over the case for years: gonadal mosaicism in a parent. If the mutation had arisen in germline precursor cells, then it could be transmitted repeatedly to offspring without affecting the parent's body in visible ways. No direct test could confirm this hypothesis. Human germ cells cannot be sampled in the way blood can, but the weight of probability pointed in this direction. In genetic counseling circles, the Darrow case became a reference point. Counselors often encounter families asking about recurrence risks after a child is born with a vascular mark. Traditionally, they could reassure parents that most marks are sporadic and unlikely to recur. But the Darrow siblings showed that exceptions exist, that even lesions usually tied to isolated mosaic events can repeat when gonadal mosaicism is present. Counseling guidelines incorporated the case as a reminder to present probabilities with caution, to emphasize that rare does not mean impossible. The broader research community drew on the Darrow data to refine epidemiological models. Population registries had already suggested that familial clustering of vascular lesions was more common than once assumed, though still rare. The Darrow record provided concrete evidence to anchor these models. A family in which every sibling bore the same congenital mark, stable over decades, with molecular data linking the lesions to identical mutational pathways. Statisticians recalibrated recurrence probabilities accordingly, noting that while the odds of such a pattern remained low, they could not be dismissed entirely. While science debated probabilities and mechanisms, the siblings themselves were simply living. Each mark became part of personal identity, visible but not pathologic, a shared trait like eye color or bone structure. In adolescence, visibility sometimes raised questions from peers, and in adulthood, the marks were acknowledged as enduring features, neither concealed nor highlighted. For the family, the shared presence of the marks became a form of recognition, a visible reminder of kinship. Their constancy made them familial emblems rather than medical curiosities. The long-term psychosocial dimension was not lost on clinicians. They observed that visible marks, even when medically benign, intersect with social identity. The Darrow siblings' adjustment illustrated how families and individuals normalize features that, in a clinical setting, become objects of investigation. This balance between scientific scrutiny and lived normalcy was one of the enduring lessons of the case. It reminded practitioners that observation must coexist with respect for personal experience, and that visible anomalies are lived realities as much as they are biological data. Academic reviews increasingly cited the Darrow case as a model of longitudinal study. Dermatology texts referenced it when discussing congenital vascular malformations, distinguishing them from hemangiomas and other proliferative lesions. Genetics texts used it to illustrate gonadal mosaicism, an elusive mechanism rarely documented with such clarity. Counseling manuals cited it as a case study in recurrence risk communication. Across disciplines, the family's decades of cooperation left a lasting footprint. The parents' role in this long narrative was also recognized. From the beginning, they had agreed to participation in studies, follow-up visits, and sample collection, understanding that certainty might never come. Their consent enabled the preservation of tissue that became valuable years later, when new technologies could extract data from it. Their openness created the conditions for the Darrow case to move from a local anomaly to an international reference. It was a partnership between family and medicine, one built not on promises of answers but on trust that contribution itself was worthwhile. By the time the siblings reached adulthood, the case no longer resembled the mystery it had at the start. The mechanisms were clearer, though not proven with absolute finality. Mosaic mutations in vascular genes had been detected. Gonadal mosaicism in apparent explained recurrence. The lesions had persisted without complication, demonstrating stability over time. The Darrow record now stood less as an unsolved puzzle and more as a complete illustration of how biology can inscribe itself on the body's surface and remain unchanged across a lifetime. And so, in their 20s, the Darrow siblings carried forward marks that had become both personal features and scientific evidence. They did not change. They did not resolve. They remained constant, visible signs of an event that occurred long before birth, signs that tied together family identity and medical inquiry. Their presence across multiple siblings offered researchers a rare window into mechanisms of inheritance and development, a window that remained open not through dramatic breakthroughs, but through the patient accumulation of detail, year after year. The Darrow family had given medicine one of its longest, clearest views into the life course of congenital vascular malformations. For clinicians and researchers, the marks were evidence of mosaicism, of gonadal transmission, of recurrence beyond probability. For the siblings, they were simply part of being Darrow, unchanging, familiar, enduring. By the time the Darrow siblings entered adulthood, the attention surrounding their case had expanded beyond the walls of their original hospital. What had once been a small series of clinical observations in one pediatric dermatology unit had grown into a record cited internationally. The years of photographs, biopsies, and molecular analyses were no longer filed away as an unusual curiosity but were instead drawn upon in lectures, reviews, and training manuals. The Darrow family had unwittingly become part of the scaffolding of medical knowledge. In dermatology, the images of the Darrow siblings circulated frequently. They showed with striking clarity how congenital vascular malformations differed from infantile hemangiomas. The Darrow lesions never receded, never proliferated in rapid bursts of growth, and never resolved with the passage of time. They held their form with an almost architectural stability. When medical students or young physicians saw these images projected on lecture hall screens, they were shown not just as anonymous photographs but as living examples of a principle. What appears at birth can persist across decades without alteration. The Darrow record became proof in images, more persuasive than any definition in a textbook. In genetics, the family's contribution carried a different weight. For decades, gonadal mosaicism had been discussed as a mechanism that could explain recurrence of traits thought to be sporadic. Yet clear, longitudinally documented cases were rare. The Darrow siblings filled this gap. Their preserved biopsy material, revisited with high-depth sequencing, confirmed mosaic mutations in vascular signaling pathways. Each sibling's lesion bore the same mutational signature, reinforcing the conclusion that a parent's germline carried the mutation in mosaic form. At genetic symposia, the case was presented as one of the cleanest demonstrations of this elusive mechanism. It reminded researchers that inheritance could operate in more subtle ways than dominant, recessive, or polygenic patterns alone could capture. Epidemiologists also drew upon the Darrow data. Familial clusters of vascular anomalies had appeared in registry studies, but most lacked detailed follow-up. The Darrow family stood out because their record stretched unbroken from birth into adulthood. When incorporated into pooled analyses, their recurrence patterns slightly shifted recurrence estimates, helping refine counseling statistics. For families anxious about the possibility of marks appearing in multiple children, The Darrow data offered a reference point that counselors could use to balance reassurance with honesty. The existence of the case prevented overconfidence in claiming, "It cannot happen again." This spreading influence raised questions of ethics as much as of science. Medical historians and ethicists examined the Darrow record as an example of sustained consent across decades. Many historical case studies were built on data collected without long-term follow-up permissions. In contrast, the Darrow family had been engaged at every stage. As the siblings reached adulthood, they were directly asked if they wished to continue participating, and they gave their own consent to the ongoing use of their images and samples. This continuity was rare. It made the case not only scientifically valuable but also ethically instructive, showing how long-term collaboration with families could be done transparently. The siblings themselves now had the ability to read the journals that cited them. They could see their case referenced in dermatology textbooks and genetic counseling guidelines, though always anonymized. Their marks, once simple features of their skin, had become material for scientific demonstration. They lived ordinary lives, studying, working, forming relationships, but they carried with them the knowledge that their bodies were also embedded in the progress of medical science. For them, the experience was neither glamorous nor invasive. It was simply the continuation of what they had always known. Annual visits, careful photographs, occasional requests for new samples. It was part of their family rhythm, as natural to them as birthdays or school photographs had once been. The research community continued to build upon the foundation their case provided. As computational models of mosaicism became more sophisticated, the Darrow data were fed into broader genomic databases. Their lesions, when analyzed against hundreds of other cases, showed the same mutational pathways as sporadic anomalies. What made the Darrows unique was not the mutation itself but its recurrence across siblings. This reinforced the conclusion that the difference lay not in biology's mechanisms but in inheritance's hidden routes. The Darrows became the living example that anchored theory to evidence. Medical training programs began to rely on the case more explicitly. Students studying dermatology were shown side-by-side images. Typical hemangiomas resolving over time contrasted with Darrow lesions persisting unchanged. Genetics courses cited the Darrows when explaining mosaicism, using their case to illustrate the limits of prediction. Epidemiology courses pointed to them as a reminder that rare does not mean impossible. The family's contribution had become embedded in curricula, shaping the education of physicians who would never meet them but would learn from their record. The siblings themselves occasionally encountered their own story indirectly. A medical student might mention in conversation that they had read about a family in which every child had the same birthmark. The siblings would recognize themselves in the story, though no names were ever given. They remained anonymous in the literature, but they knew that their lives had been translated into lessons read by thousands. Their marks were visible to anyone they met, but only those in medicine knew the wider narrative. To the rest of the world, they were simply people with distinctive skin features. To science, they were a key reference point. The parents, too, understood the shift that had taken place. What had begun for them as anxious questions in a pediatric clinic had evolved into a long collaboration with researchers. They had lived to see their children grow healthy and independent, carrying the same marks but no medical complications. They had also lived to see those marks written into the language of journals and textbooks. Their choice to allow follow-up had become part of medicine's collective memory. By the time the Darrow siblings were firmly established in adulthood, their case had transcended its origins. It was no longer confined to the family or the clinic where it began. It had become a tool of education, a reference in genetic discourse, a data point in epidemiological models, and a case study in ethical collaboration. The Darrows remained themselves, living ordinary lives, but their existence had also been translated into the infrastructure of medical knowledge. As the Darrow siblings entered their 30s, a new question emerged that had never been part of the original study. What would happen when they began to have children of their own? The recurrence of the marks across all 4 siblings had been documented with remarkable precision, but medicine had not yet seen whether the pattern would carry into a second generation. This uncertainty placed the Darrow case once more at the intersection of clinical observation and personal life, blending the scientific record with intimate family decisions. The siblings had grown up with medical attention woven into their lives, and so genetic counseling was almost instinctive when they reached the stage of family planning. Unlike most prospective parents, they did not need to be told about recurrence risk. They had lived it firsthand. For years, specialists had explained that while gonadal mosaicism in a parent was the most likely explanation for their shared marks, the transmission to grandchildren was not a certainty. If the mutations were confined to germline cells in the first generation's parent, then the second generation might not inherit them at all. On the other hand, if one of the Darrow siblings carried the mutation systemically in a subset of germ cells, then the marks could appear again. It was a statistical unknown that could only be resolved by experience. The first pregnancies within the family were followed with both anticipation and careful medical observation. Obstetric teams were aware of the family history and arranged for early neonatal examinations. When the first child was born without any visible lesion, clinicians noted it with interest but also caution, recognizing that absence in one child did not establish a pattern. When the second and third children were also born without the marks, questions began to shift. Perhaps the recurrence had ended with the original sibling group. Perhaps the parent who carried the gonadal mosaicism had passed on only a limited risk that did not extend into subsequent generations. Still, medicine could not dismiss the possibility entirely. Reports in other families showed that gonadal mosaicism could persist unpredictably across generations. For the Darrow grandchildren, each newborn examination became part of a broader record. Physicians took high-resolution photographs of the skin at birth and again at early milestones, just as had been done with their parents. Each clear finding was added to the Darrow archive, expanding the family record into a second generation. The absence of lesions was itself data, just as important as their presence. As years passed and more children were born, the trend became clear. None of the grandchildren carried the birthmarks. For the Darrow siblings, this brought a sense of relief, as the recurrence they had lived with did not seem to extend into the next generation. For clinicians, it was confirmation that gonadal mosaicism in one of the original parents had been the most likely mechanism, and that it had not translated into systemic inheritance through the siblings themselves. The second generation's clear skin added weight to the conclusion that the family's unique pattern had been the product of a single parental mosaic event, rather than a dominantly transmitted mutation. Outside the family, the Darrow case continued to ripple through medical education and public awareness. Medical students who had studied the case in their textbooks sometimes encountered references to the Darrow pattern as shorthand for recurrence in siblings without recurrence in offspring. It became a teaching phrase, not official nomenclature but a colloquial marker among those who used the case to illustrate gonadal mosaicism. This informal recognition gave the Darrow family a kind of invisible presence in clinical culture, their story known by thousands of physicians worldwide even though their names were never revealed. In counseling clinics, the Darrow example was raised when families sought advice about recurrence risk for vascular anomalies. Counselors explained that while most cases were isolated, families like the Darrows showed that recurrence could happen. They also pointed out that the Darrows' next generation had been unaffected, offering reassurance that risk did not always extend indefinitely. The balance between caution and comfort was delicate, and the Darrow case provided a real-world framework for striking it. Families who worried about the future of their children were given not only abstract probabilities but also an anonymized story of a family who had lived through similar uncertainty. The research dimension of the Darrow legacy deepened as biobanks expanded. With the grandchildren's consent, even their DNA was sometimes included in comparative studies, creating a multigenerational dataset. Sequencing revealed no systemic mutations, aligning with the absence of lesions. Their genomes served as control data against their parents' preserved tissue, further confirming the mosaic nature of the original marks. The Darrow archive thus extended across 3 generations: the original parents, the siblings with marks, and the grandchildren without. This made the case not only rare but uniquely complete, offering geneticists a full arc from the emergence of mosaicism to its resolution in descendants. Meanwhile, the Darrow siblings reflected on how their story had traveled beyond them. In classrooms and conferences, their marks were still being projected on screens and dissected in academic papers. They knew that their case had entered the medical canon, but in their everyday lives, the marks were simply part of who they were. Their children, unmarked, grew up without the layer of medical scrutiny that had accompanied their own childhoods. For the Darrows, this was a quiet relief. Their children could grow without the annual photographs, the clinic visits, and the questions that had shaped their early years. The scientific legacy persisted, but the lived reality had shifted toward normalcy in the second generation. The Darrow case also began to filter into the broader public sphere through medical writing aimed at general audiences. Articles in science magazines occasionally mentioned an unnamed family in which all the children had shared the same birthmark, sparking curiosity among readers who wondered about the strangeness of inheritance. The story, stripped of names and identifying details, became an example of how biology can appear mysterious while still following identifiable mechanisms. For the family, anonymity was preserved, but their experience had entered public imagination, blurring the boundary between private history and collective knowledge. In academic circles, debates continued about how best to classify such cases. Some argued that the Darrow pattern should be described formally as an example of familial recurrence due to parental gonadal mosaicism. Others suggested that too rigid a label risked oversimplifying what remained a rare and complex event. Regardless of terminology, the Darrow case remained the most detailed documented example. Researchers cited it when writing reviews on mosaicism, congenital vascular anomalies, and recurrence risk. Its weight grew not from drama or rarity alone, but from the sheer detail of its documentation, stretching across decades and generations. For the grandchildren, the legacy was both distant and immediate. They had no marks of their own, no medical reason to be followed, yet they grew up knowing that their parents' skin carried features that had once been the subject of international discussion. They were aware, in fragments, that their family had contributed to medicine in a way few families did. Yet their lives unfolded without the constant observation that had shaped their parents' childhoods. The Darrow archive belonged to them only as history, not as present experience. And so, as the Darrow story entered its second generation, the case continued to grow, both as scientific evidence and as lived memory. The grandchildren's clear skin added new dimensions to the archive, while the siblings' marks remained as visible as ever, unchanged but reframed by time and by the expansion of the family line. Medicine moved forward, carrying the Darrow record with it, and the family continued to live with the dual reality of being both ordinary individuals and enduring figures in medical literature. By the time the Darrow siblings entered middle age, their lives were firmly established, and the marks that had once defined their earliest medical experiences remained exactly as they had always been. Visible, unchanged, part of them. For clinicians, the fact that these lesions had now persisted into the fifth decade of life was invaluable. Very few congenital vascular malformations were followed so consistently, across so many years, with photographic evidence at every stage. The Darrow case had evolved from a clinical curiosity to an archival cornerstone, a dataset preserved and referenced in ways that stretched beyond the family itself. Medical archives had grown in sophistication since the days when their first photographs were taken on film and stored in paper files. Now, the Darrow record existed in digitized form, carefully anonymized and integrated into dermatology and genetics databases. Researchers who accessed these archives often did not even know the name Darrow. They saw only anonymized identifiers, case numbers, and image series, but the continuity of the files was unmistakable. Few other records displayed such clean longitudinal consistency. For meta-analyses comparing long-term outcomes of vascular malformations, the Darrow siblings files were among the most cited. As the digital age advanced, these archives became part of broader cohort studies. Large consortia brought together cases from across continents, pooling thousands of examples of congenital anomalies into searchable databases. Within these massive datasets, the Darrow record stood out because of its unusual recurrence in siblings and its unusually complete follow-up. When algorithms searched for longitudinal completeness, the Darrow files consistently appeared at the top. They were used to validate automated image recognition systems that trained on dermatological features, teaching computers to distinguish between transient hemangiomas and persistent vascular malformations. The siblings' skin, once photographed in clinical rooms for physicians alone, now trained machine learning models that would one day assist in clinics worldwide. The ethical dimension of this archival presence deepened with time. Medical ethicists raised questions about how long such data should be stored, how it should be used, and whether the family should have ongoing control over it. The Darrow siblings, now adults with decades of participation behind them, were again consulted. Their responses reflected the balance they had always maintained. They did not seek to erase the record, nor did they resist its use, but they insisted on transparency. They wanted to know how their data was being used, whether in teaching, in machine learning, or in international research. Their insistence became a case study in its own right, used in bioethics courses to illustrate the principles of continuing consent in long-term medical research. Meanwhile, their children, the second generation who had been born without the marks, were now old enough to understand the role their family had played in medical science. They had grown up without annual clinic visits or photographic sessions, but they were aware of the archive. Occasionally, they encountered references to their family's case in academic writing, described in anonymized terms. To them, the story was part of family history, like old letters or photographs, but its reach extended into classrooms and journals they might never read. They inherited not the marks but the knowledge that their family's experience had become part of the permanent fabric of medical literature. The persistence of the Darrow lesions into middle age also added fresh data for clinicians. Many vascular malformations change with time, thickening, darkening, or developing nodular components. The Darrow lesions, however, remained stable in texture and color. Their lack of progression became another rare point of reference. When physicians advised patients with similar marks, they could cite the Darrow record as evidence that not all lesions worsened with age. The Darrow siblings became examples of benign persistence, offering reassurance to families who feared disfigurement or deterioration. Beyond dermatology, the Darrow case also influenced how medicine thought about memory and record-keeping. Medical historians began to write about the case not only as an example of mosaicism but as a study in the value of longitudinal follow-up. The Darrows illustrated what could be achieved when families remained engaged across decades. Their story became an argument for building better infrastructure to support long-term observation, reminding researchers that true understanding often required patience measured in generations. By the time the siblings were in their 50s, their case had become more than a clinical record. It had become a touchstone in the history of medical science. Their photographs were still projected in lecture halls. Their genetic data still appeared in databases. Their names were never revealed, but their presence lingered wherever mosaicism, recurrence risk, or congenital anomalies were discussed. They were ordinary people who had become extraordinary data points, not by choice or by design, but by the persistence of marks on their skin and the persistence of the physicians who followed them. The siblings themselves continued with their lives in ways that were outwardly unremarkable. Careers, families, and communities occupied their days. Yet woven into those ordinary lives was the knowledge that their existence had shaped the training of physicians across the world. Students who had never met them nonetheless knew their images, their case, their marks. The Darrows had become part of collective medical memory. Their story retold each time a counselor explained recurrence risk or a lecturer described mosaicism. And still, the marks endured, unchanged, constant, reminders of an early event in cellular division that had left a trace on the surface of the skin and a record in the annals of medicine. As the Darrow siblings grew older, their lives settled into rhythms that had little to do with medicine. Careers deepened, children grew, and grandchildren began to appear. The marks, present since the first day of their lives, remained constant companions, neither fading nor worsening, neither burdensome nor forgotten. They were simply there, as much a part of the Darrows as eye color or voice. By now, the siblings had lived with them for 6 decades, and the world around them had changed more than the lesions ever had. In the medical community, the case still held quiet prominence. The digital archive expanded once more, as updated photographs were taken in later years, documenting how the marks appeared on aging skin. Geriatric dermatologists noted the importance of this. Few congenital lesions were observed into old age with such precise continuity. The Darrow series had become one of the rarest of records, showing persistence not just through childhood and adulthood but now through the late decades of life. The images, lined with the texture of age, carried a gravity that the earlier photographs could not. They demonstrated endurance across the human lifespan. Bioethicists occasionally revisited the case in their writing. They pointed out that few families had ever given such prolonged consent, across generations, for their data to remain in circulation. The Darrows, through steady willingness rather than deliberate ambition, had shown how transparency and respect could preserve trust over a lifetime. For students of medical ethics, the Darrow record became not just a clinical lesson but a model of partnership between science and family. Meanwhile, in conference halls and lecture rooms, the case was still cited. A new generation of students, born decades after the Darrow siblings, encountered the photographs in slideshows and textbooks. They learned to identify vascular malformations by seeing the unchanged lines on Darrow's skin. They heard about gonadal mosaicism not only as theory but as something proven, demonstrated, confirmed in the lives of one family. For those students, the Darrows were not names, not people they would ever meet, but a thread of knowledge woven into their training. Within the family itself, the archive was less present. The siblings rarely spoke of the medical record anymore, except when contacted for updates. Their children and grandchildren lived without the marks, without the clinic visits, without the strange attention that had once followed their parents. For them, the story belonged to history. They knew, in a distant way, that their family's experience had shaped science, but they carried no reminder on their own skin. The case lived more in books and databases than in their daily lives. As the siblings reached their later years, the marks were still visible. The same shapes that had been photographed in infancy, unchanged in size and location, now resting on skin weathered by time. Physicians who saw them during routine checkups sometimes paused, aware of the case they represented, aware that they were standing in the presence of a living archive. For the Darrows, it was ordinary. For medicine, it was extraordinary. The record, stretching across 7 decades, remained intact, from birth, through youth, into adulthood, into old age. No resolution, no fading, no disappearance. Just persistence, in the skin, in the data, in the memory of a discipline that had watched, recorded, and learned, and so the Darrow story settled into permanence. The marks endured. The archive endured. The family endured. Medicine, in turn, carried their presence forward, not as names, not as individuals, but as a case that would never be forgotten. A thread of continuity woven quietly into the history of human knowledge. The Darrow story may close here, but countless others wait to be uncovered. If you'd like to continue exploring with me, join me at Eternal Obsidian.